Today we are going to discuss an ELISA technique procedure in a step-by-step way. ELISA Assay is short for Enzyme-Linked Immunosorbent Assay. It is a technique used to quantitatively detect specific proteins in a complex mixture such as cell lysate, blood sample, and more. It is widely used in research because of the versatility and potential antigens it can detect and quantify. The technique utilizes the specific interaction between antigen and an antibody. To detect the presence of the antigen, a detection system will be employed that is either conjugated to the primary antibody or secondary antibody that binds to the primary antibody. A detection system is used to quantify the amount of antigen present in the sample. The intensity of the signal will be directly proportional to the amount of antigen captured in the microplate. Widely used detection systems include conjugation of a fluorescent tag or an enzyme. A substrate will have to be introduced to the enzyme by the end of the assay, with the product produced by the enzyme being measured. A commonly used detection system is alkaline phosphatase. PNPP is a substrate of alkaline phosphatase, and the system forms a yellow soluble product, p-nitrophenol. The color can then be detected and quantified by a spectrophotometer. A 96-well or 384-well polystyrene microplate is normally used for the assay. Polystyrene is composed of aliphatic carbon chains with intermittent benzene rings, which makes it very hydrophobic. It can form hydrophobic interactions with the nonpolar protein residues and allow the protein sample to passively absorb onto the well walls. The color of the polystyrene microplate is chosen based on the type of signal being detected. For example, clear microplates are used for colorimetric signals. The first step is coding and capturing samples onto the microplate well. Direct capture relies on the passive absorption of sample onto the wells, following by the addition of free primary antibodies. The sample is usually dissolved in an alkaline buffer and directly coated onto the proteins. This method is nonspecific, as everything in your sample will be coated onto the well. In indirect capture, the wells are coated with antibodies that selectively bind to our target antigen. The first problem we may encounter during the capture step is the improper orientation of the protein sample, denaturation of protein, or binding contamination. Capture antibodies can be used to help with the proper orientation of proteins. Coating the microplates with polymer can also increase passive absorption of our samples onto the well. This increases the binding of primary antibodies to our sample. After our sample is settled in the well, the coating solution is removed. Then, we will need to perform plate blocking. Plate blocking is the addition of irrelevant protein to block off the unused surface binding sites of the well. This will help reduce background noise during the detection step. After blocking, the microplate needs to be washed to remove unbound material. The sample will now be ready for the detection step. Similar to capture, the detection step can be broken down into direct detection and indirect detection. Direct detection is when the primary antibody is directly conjugated to a detection system. Indirect detection is when the primary antibody is recognized by a secondary antibody. There are several advantages to indirect detection, because the conjugation of an enzyme or fluorescent tag to the primary antibody may affect its immunoreactivity. Using a second antibody can preserve the immunoreactivity of the primary antibody. The use of a secondary antibody as a detection system can also result in signal amplification, because multiple secondary antibodies can bind to one primary antibody. A disadvantage to having a secondary antibody is that cross-reactivity might occur, but this is normally eliminated through using antibodies from different host species for the primary and secondary antibodies. After incubation with the detection system, the well is ready for reading. For the ELISA assay, an enzyme is normally used for the detection system, so its substrate needs to be added. The reader will have a color product that can be measured by a plate reader. To read and analyze results, a standard curve needs to be made with known antigen concentrations plotted against their readouts. For example, in this figure, known concentrations of IFN gamma are plotted against absorbance. The readout of our assay can then be compared to the standard curve to determine the concentration of IFN gamma in our sample. There are 4 different types of ELISA assays: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. In direct ELISA, antigen is directly bound to the microplate well and is bound to a primary antibody that is conjugated to a detection system. In indirect ELISA, antigen is directly bound to the bottom of the microplate. A primary antibody is used to capture the target antigen, and a secondary antibody conjugated with the detection system can bind to the primary antibody. As mentioned before, multiple secondary antibodies can bind to one primary antibody, which leads to signal amplification. In sandwich ELISA, the antigen is indirectly captured and indirectly detected. The antigen will need to have two different target epitopes for the binding of the primary antibody and secondary antibody. The primary capture antibody is boated on the microplate to capture our target antigen. The secondary antibody is conjugated to a detection system and recognizes an antidote that is different from the capture antibody. This method is highly specific and sensitive. However, it does require more optimization to identify the antibody pairs. The competitive ELISA assay is used when antigen is small or when it only has one epitope. The microplate will be pre-coated with secondary antibodies. Besides the addition of primary antibodies and sample, purified antigen labeled with a detection system are also involved in this assay. The purified samples labeled with the detection system will compete with our sample to the binding of primary antibodies, which eventually binds to the secondary antibody coated on the well. The signal output will be inversely dependent on the sample concentration.